Okay. Thanks, Gabriel. You can see my screen?
Yes.
Okay, great. So, well, I want to thank the organizer at Syntica to let me present some of my data here that I acquired during, like you said, my PhD thesis and then my postdoc. And I'm excited to talk about sort of the DEXA system and the different systems that I've used in the past and how they allowed me to sometimes make like pivotal change in my science and structural change and conceptual change in my ideas and things like this. So yeah, so the outline of my, sorry, uh, of my presentation, like I said, I'm going to talk about dual X-ray absorptiometry, so DEXA in skeletal studies, and then I'm going to go into the data that I acquired during my PhD. So it's about the IGFP before knockout mice and their skeletal phenotype and beyond, thanks to DEXA. And then I'm going to talk about the preliminary results of our ARC4FC treatment in mice. That's something I'm currently doing during my postdoctoral research and now my instructorships at Harvard School of Dental Medicine. And then I'm going to compare two DEXA systems that one that we have here at HSDM and another one that we have. And I'm going to add a little more DEXA system that I found around the Boston area. So I hope you can bear with me. So I want to talk about skeletal phenotyping in mice. So these are micro-CT images of the distal femurs, so in aging mice, in males and female mice. And the reason why I put this is I wanted to give the audience a chance to appreciate the radical changes that mice go through during their aging in terms of the architecture of the bone. You can see the quantity of mineral tissue here is really changing over aging. You can see they have mice usually reach big bone mice between 8 and 14 weeks of age, depending on sex and strains. And then they start losing bone rapidly, just like we do. And by, a year of age, the bones are quite osteoporotic. You can see there's no more cancellous bone in the marrow cavity here. And so when bone scientists, we're thinking about bone mass and skeletal phenotyping, we need to be able to appreciate all of those differences. So not necessarily just the bone quantity or bone mineral density outside the bone, but also the macroarchitecture inside the bone, because all of this together is going to create bone strength. So our resistance to fracture, basically, which is what the skeleton is important for. So the sort of gold standard to look at all of this together is micro-CT, those images that I'm showing right now. The issue with micro-CT is that, as you can see, I have to isolate the bones, so sacrifice mice, take the bones out, fix them, process them, and scan them, which takes a long time. And there's a lot of limitations for micro-CT for this. So it's not necessarily something I can do longitudinally. So I would have to raise many mice in order to do this. And it's also very expensive to do and very time consuming. So they have invented in vivo models since, but they're not really fast, or at least not as fast as DEXA. And so I plot those, this is BVTV, so it's the quantity of bone found inside the marrow cavity here over time. And like I was telling you, it really changes drastically over the age of the mice, and you can appreciate that here. So I think I made the point that all of this together is really gonna affect the structure of the bone. And so in order to be looking at this, we really needed to come with systems that were allowing us to look at changes in living mice over time. So this is the micro-CT limitation that I just talked about. And I think I just went through this. So, they require me to sacrifice mice to isolate the bone. They're very slow. It takes us days worth of work to process an entire cohort. So the other limitations that I wanted to list is the measurement threshold are set by the users. So in micro-CT, you get slices of all of your bone, right, longitudinally or whichever way you set it. And then a user has to go in and contour and set a segmentation threshold. So basically telling the machine what area to look for and what do you consider bone. So that is going to be set by me or by someone else. And obviously that's going to be affecting the quantification result at the end. So that is a limitation because, it's difficult to then compare across studies. If you've used different segmentation, you actually cannot. It's also, like I said, expensive. So that is sometimes a limitation, especially when you have really large number of mice because you're trying to look at a small effect or something like this. It will be very expensive to conduct microCT for all of those. And again, they started to come out with in vivo CT, so that's available. So you don't necessarily have to sacrifice your mice, but those are even more so expensive, very time consuming, and sort of logistically they're difficult to set up in a environment. But they are coming. Most people have them. It's just not something you routinely do on large cohorts of mice at multiple time points. So that's why I said they invented the dual X-ray absorptiometry, and we have them. And it's the DEXA basically use two X-ray beams at different energy, and they measure bone mineral density that way. But they measure also other parameters. So that's going to be important for me. Obviously, I talk about bone mineral density because I'm a bone biologist, but we started to get interested into other parameters too. And so these are just examples of DEXA here because they're also used in clinics. And actually in clinics, they're one of the most routinely used scans, especially to measure bone mineral density, because obviously you cannot necessarily do microCT in humans because you cannot isolate their bones, right? So so you do DEXA, it's pretty fast, and then they have indices and sort of numbers to try to chart those humans and tell them if they have osteoporosis or issues with their bone mineral densities. But we also have DEXA, obviously, for small animals. And I put a picture of the PIXImus here, which was the first system I used when I was doing my PhD with Clay Frozen in Maine. So that's why I put that one here. So the strength of the X-ray, it's non-invasive. So like I said, you don't have to necessarily sacrifice your mice to use it. There's a minimal dose of radiation. And that is important because, again, whether you're at an institute or clinics or things like this, setting things up with radiation can be very limiting because you need to have proper sort of rooms and signage and approvals and things like this. The DEXA here was set up like we got it within a month because it's such a low dose of radiation that it doesn't really cause any issues. The scanning time is quite fast, especially compared to microCT. We're talking, seconds or minutes over hours, sometimes days. So it's very fast and it's cheap, relatively speaking. Sorry. So those are the strength of the DEXA. So like I said, I want to start with the PIXImus system because it's the first one I've used back then. And so the PIXImus system is here. And this is just an image I took of my data back then. And so this is what the mice looks like when you scan them. And so it's a portable unit. As you can see here, it's pretty small. It's only about 60 pounds. So, portable is in quotation mark, right? I don't think I would want to carry 60 pounds with me every day, but you can move it pretty easily. It's using a stationary anode X-ray tube, and that generates a cone beam. And it's, like I said, using very low X-ray energies, so it's pretty safe. And it has an ultra high resolution pixels. And so it can achieve contrast in extremely low density bone and for tissue comparative studies. So that's important for us because obviously we have mutants or mice sometimes that have very low bone density, and we need to be able to detect this. So here you see the image of the skeleton of the mice that is outlined in yellow and the whole mouse outlined in blue. And you see the little red that is at the little red sort of rectangle at the femoral site. That's because we were looking at overall bone mineral density, but also femoral, so site-specific ones. And we use the femur routinely for this. And the green sort of blacked out circle you see, it's just because we exclude the head when we are looking at bone mineral density in the mice, because obviously teeth are hypermineralized, so that will skew your data, but there's also a lot of fat tissue and differential tissue in the craniofacial elements that might skew your data differently. So in order to account for that, we just discard that on all of our samples usually. But I can see how other people, maybe, I'm at a dental school now, so obviously people actually look at the teeth, so they don't exclude it. So that's depending on your setting. Was just my setting for my studies. So now I'm going to go into my studies. So that, like I said, I'm going to talk about my PhD thesis where I used that PIXImus. So I did my PhD thesis around IGF-1. And so IGF-1 is insulin-like growth factor. And some of you may know that this is one of the circulating hormone that's basically regulating all of our growth. And this works because hypothalamus can secrete, hormones that are going to act on the pituitary to release growth hormone. I would assume that everyone knows growth hormone is obviously important for growth, but it doesn't act really directly on all of the tissues. What it does is that it goes to the liver and acts on the liver cells and make them release IGF in the circulation. And IGF is then going to carry out all of those actions on the metabolism and sort of the anabolic growth of tissues. And it's especially important for adipose tissue, bone, and skeletal muscle. So we are bone biologists, so we were very interested into IGF-1, right? And so uh, like I said, bone biologists are interested into IGF-1 because IGF-1 can act on mesenchymal progenitors. Mesenchymal progenitors are found in the mesenchyme tissue during embryogenesis, and they are the stem cells that are going to self-renew here and also going to give rise to adipocytes. So cells of fat tissue, chondrocytes. So those are cells that form our cartilage and ellagens during embryogenesis. So they're very important. And then they form the growth plate and so on and so forth. They form a lot of craniofacial elements as well. And then osteoblasts, which are the cells responsible for laying down the matrix that's going to get mineralized in bone. So they are the bone forming cells. And as I've indicated here, insulin-like growth factor 1 is important to regulate not only the self-renewal of mesenchymal progenitor, but the differentiation and the function of all of those cells. So it really is like a master regulator of the mesenchymal transition and mesenchymal biology, the mesenchymal stem cells biology. But you, you know, it cannot act like this, do all of those things, because then how would it pick one pathway versus the other? That's when there's a layer of regulation, an extra layer of regulation that comes on top of IGF-1 in the forms of binding proteins. So this is what I label here, the IGF binding proteins. And, I say that they're bind to IGF because that's what that's what they do. And in circulation, you found IGF bound usually to these proteins. So there are multiple IGF binding proteins, and they can sort of regulate if IGF can act or not. And here I listed them. There's 6 IGF binding proteins, and I listed some of their known function and activity in different sort of tissues. The IGF binding 1 is important for glucose metabolism. IGF binding protein 2 is important for fat and bone. And we had someone else in the lab at the time that was working with IGF binding protein 2, Vicky DeMambro. And so she had done beautiful work already with this. So that was during my thesis, I was very excited to work on IGF binding protein 4, which I just highlighted here, which was thought to be important for growth and in both the adipocyte and the osteoblast, because both of those cells could produce IGF-BP4, but not much else was known about IGF binding protein 4. So I will go into what was known at the time. So it was thought to inhibit the action of IGF in almost all in vitro models. So when they studied it in different cell lines and primary cells, they saw that IGF actions were blocked when you were treating with IGF-BP4 for bone cells, so different osteoblasts and also some of the mesenchymal progenitors, in vascular smooth muscle cells, and in colon cancer cells. So it seems to really be blocking IGF-1's action, so probably inhibiting its binding to the receptor for IGF. And then in vivo, it was a little more complicated at the time. So they had models of overexpression of IGF-BP4 and in bone specifically, and that reduced bone formation. So that was sort of consistent with what we were saying, blocking the actions of IGF. However, when they injected IGF-BP4 systemically in mice, that induced bone formation. So that was really discordant with what was just seen before. And then surprisingly, the knockout mice had a growth delay, right, which was expected, but unfortunately, that knockout was generated on a mixed background. So it was a little difficult to say if that was specifically due to IGF-BP4 or more sort of systemic effects with other genes, because when we study mice, we like to have genetically pure backgrounds, so inbred backgrounds strains, so that we are show that no other gene can be involved in our knockout models, really directly causing the phenotype, but it's due to the loss of the specific gene we're looking at. So although that was really useful data, this mixed background study, we wanted to do it. So we had a hypothesis that IGFBP4 was regulating postnatal growth, and specifically by affecting the skeletal acquisition. So we thought that there was going to be a huge bone phenotype in the knockout mice. So we took the knockout mice, and before my time, they had put them on the C54 Black 6 background. So I was really lucky to join the lab at that time because they have, I think one of them was Vicky, had done all of the initial work to do this. So that was a great time to join the lab. And so we took the IGFBP4 now knockout mice, and I first checked that obviously IGFBP4 was deleted in the bones of those mice. And you can appreciate here that it was deleted in both females and male mice. So they were expressing any IGFBP4. What was interesting is that at 8 weeks of age, those mice had decreased body weight, both in the female mice in gray and male mice in white. And then when I look at 16 weeks, that body weight decrease was persisting, and the body length was also decreased, indicating that there was an overall growth retardation in the IGFBP4 knockout mice carried from 8 weeks to 16 weeks of age, which, as you remember, was consistent with the previous finding on the mixed background. They had reported a growth delayed in mice, specifically in females. So that was very consistent with what we have. So the model was working pretty well. So then obviously we did DEXA because I said our hypothesis was looking at bone mineral density. So this was what this was. And so these were the parameters that we measured. We saw that, and I have the 8 and 16 weeks and the female in gray and the male mice in white. So as you can appreciate, at 8 weeks of age to start, there was a trend towards a decreased bone mineral density, but really nothing very significant at all. But then at 16 weeks of age, the females had a significant decrease in both areal and bone mineral density and bone mineral content, while the males did not have any change. So it seems like between that period of growth from 8 to 16 weeks, they didn't acquire bone mineral density and content in the skeleton, in their overall skeleton. I looked also, so that was overall the whole body of the mouse. Femoral, I look at the femoral BMD and BMC, and it was following similar trends as well. So then obviously I had to do micro-CT because, like I told you, this is the gold standard, and this was to look inside the bone because we cannot do that with DEXA. So after we were able to see a change in bone mineral density with DEXA, it was worthwhile investing the money to pursue it further with micro-CT. And as you can see here, micro-CT confirmed what we were thinking with DEXA. So the male mice were not, did not have a difference in bone volume over total volume, which is basically this trabecular bones that you can see on the left. And they didn't have a difference in the cortical bone too, which is what you see on the right. So that's the mid shaft area of the femur. So that's why it looks kind of like a donut. And so the females, however, had a significant decrease in both parameters, so the cortical area and the bone volume of the trabecular compartment. So that was all confirmed. And so we were getting very interested into this difference between male and female mice. And again, I was very lucky that Vicky could do ovarectomy in the lab. So she showed me how to do this, and she went and we ovarectomized those mice. So we took female mice at 8 weeks of age and we ovarectomized them to then look at their bones. And what we saw is that obviously with ovarectomy, you have a loss of estrogen. So that mimics sort of what happens in postmenopausal women, and it induces a lot of bone loss because that's what happens when you have loss of estrogen, which you can appreciate here in the first two bars. There's a significant decrease in bone mineral density and bone mineral content. And as you can see in my knockout, they're completely protected against that loss. So that was extremely exciting because now I started to think about estrogen regulation of IGFBP4, right? And so I ended up looking at that, and it turns out that IGFBP4 is more expressed in females, and it seems to be under regulation of estrogen. So I'm not going into all of that data, but there's publication out there indicating this. And so at that point, I had the beginning of a model, basically, where in the osteoblast estrogen could act and, like I said, increase the production of IGF-BP4, which was then regulating IGF-1 bioavailability locally in the bone. And so that could really regulate the differentiation and the mineralization potential of the osteoblastic cells here. And I didn't show it because just for sake of time, but we did see change in sclerostin, circulating levels of sclerostin, which is a protein produced by differentiated osteoblasts, so osteocytes that are embedded in the bone and that inhibit bone formation. And we showed that it was also different in our males and female mice in the knockout, and that seemed to be participating in this whole regulation of the osteoblast activity under the IGF-BP4 knockout mice. And I just had put the osteoclast there because just to say that those didn't seem to be really changed in our mice. So it didn't seem like we really affected osteoclastic activity very significantly. And so all of this together is obviously participating in this postnatal skeletal acquisition and leading to a normal BMD and BVTV. So when you knock out IGF-BP4 in females, you are affecting all of this system. And that's why my mice didn't have normal BMD or BVTV. And that's why I think the males, because obviously males don't have that much estrogen, especially from 8 to 16 weeks of age, that's why I didn't see a phenotype because they don't really have that system. So that was really early on, and that was all well and good, but I was, that's not enough to graduate basically. So I went to Cliff and I said, well, Cliff was saying, well, you need more mechanistic and you need to do a little more than this and dig a little deeper if you want to graduate. Cliff is really nice, he didn't say it that way, but it was clear that I needed to do a little more. So what was really nice is when we looked at the DEXA data, we went back to it, we realized that fat proportion, which is one of the parameters measured by DEXA, as well as lean mass, was significantly decreased in both males and female mice this time, not just in female mice. So, that was very interesting because now I would have, I personally would have missed that phenotype because I wouldn't afford to look necessarily at adipose tissue. Obviously, my coworkers, Vicky and Cliff would have told me to look at it, so I don't think I would have missed it because of them. But on my own, I would have certainly missed it. That was not necessarily something I was thinking of at the time. But here you can see that, like I said, the fat proportion were significantly reduced in my mice. And so I went back and I isolated the fat pads from the IGF-BP4 knockout mice. And in the inguinal white adipose tissue, the gonadal white adipose tissue inside the peritoneal cavity, and the interscapular brown fat. And you can see that there are smaller fat pads. So histologically, you can appreciate that that was confirming what we were seeing by DEXA. So that was very interesting to us. So I isolated cells, mesenchymal progenitors from those mice, because remember, the mesenchymal progenitors give rise to the bone-forming osteoblast, but also to the adipocyte, right? So we designed an in vitro assay where they were differentiated into adipocyte. So they were differentiating the stem cells from males and female mice, and then stain them with something called all-red-O. So ORO is all-red-O, and that's basically stains lipids in red. So all of these little red dots you see are lipids, and you can appreciate that there's a lot less in the knockout than there are in the wild type. Right, and that's quantified on the right. So again, confirming that this is something we see in vitro. So it's mostly like a cell autonomous phenotype. It's probably not just occurring because of a systemic effect inside the mice, but more of a local production of IGF-BP4 most likely. And so I showed that as you differentiate cells into adipocytes from 0 to 8 days, there was a huge increase in IGF-BP4 expression. So that was really confirming everything, saying that when a mesenchymal stem cell progenitor starts to differentiate into adipocyte, they express IGF-BP4, most likely to regulate the bioavailability of IGF-1 locally in their tissue. And so that affects obviously their biology, right? So one other thing I did was to look at phospho-AKT, which is the downstream activator of IGF. And here I could see that there seemed to be a significant decrease in phosphorylation of AKT, especially in the differentiated adipocyte, but also in like the earlier mesenchymal stem cells in the knockout mice. So indicating that there was indeed less IGF signaling going on. And so that's why IGF-before was inhibiting IGF in vitro here, which was consistent, like I said, with all of the other in vitro models that were published. So thanks to this, I could finally graduate. So I was really happy with the DEXA data because that really put me on the right path. And so my PhD was great. I would really do it all over again. It was really a great experience. I had great mentors during my PhD, so I could graduate. So, and of course, I could move on to HSDM, which is where I am right now. And continue studies. So when I joined HSDM at the time, they did not have a DEXA. So, sorry, they did not have a DEXA. So that was not something, I was like a little shocked by that because I thought DEXA was such a cool machine. It had allowed me to get so much data during my PhD. So I was really thinking about getting a DEXA. But before I move on to talking about the data I got during my postdoc, I think there is an audience for here that Cintika wants to do. So I will pass it on to you, Gabrielle, for this.
Thank you, David.
Yeah.
Thanks for the great discussion as well. It's very exciting. And as David mentioned, before we continue, we would like to ask a couple of quick poll questions, and the questions are the following. Just give me a second. It should be seen now. Okay, so the first question is, are you planning on attending any of the following scientific conferences? And the options are SNNMI, which will take place this month in Chicago, iSiGHT, which will also take place in Chicago. Not in Chicago, in Madison, Wisconsin this month as well. The third option being iSiGHT in Europe, which will take place in July in Porto, Portugal. BCVS, which will also take place in July in Boston, Massachusetts. WIMIC in September in Prague, Czech Republic. ASBMR in 2020, ASBMR in October in Vancouver, BC, and the final option being other. The second question we're asking is, have you used DEXA imaging in the past? And the options are the following. Yes, I currently use DEXA in my studies. Yes, I previously used DEXA in my studies. No, but I hope to add DEXA capabilities to my lab in the near future. And the final option being no. We'll just wait a few moments for everyone to answer the questions. And after that, I will hand over the presentation to David. We will just wait a few moments and we will continue the presentation. Okay, awesome. Thank you for answering the poll questions. I will now hand over the presentation back to David.
Thank you, Gabe. Okay, so I'm gonna go into the postdoctoral research I've done at HSDM. So I finished my PhD and now it's my postdoc. And for my postdoc, I wanted to look more into signaling. So like I said before, the last piece of data I'd acquired during my PhD was about IGF signaling. So I was really interested into signaling in bone. So I joined Vicky Rosen's lab, who is really focusing on BMP signaling, so the TGF-beta pathway, basically. And so here, the TGF-beta superfamily of ligand really regulates two massive pathways, the SMAD158 pathway, mostly regulated by BMPs with those receptors, the activin signaling pathways that activate SMAD2/3, and the TGF-beta, obviously, that activates SMAD2/3 as well. So those two branches, the SMAD158 and the SMAD2/3, are central regulator for bone homeostasis and bone acquisition. So we're really interested into this, but specifically, I was interested into activin signaling. And the reason is we had early data showing that activin first goes up in age significantly in the circulation from 2 to 12 months of age. And if you remember my first slide, this is really where we see this huge dramatic decrease in bone. So that was a very attractive candidate for studies. That was also shown in the bone. So if you remember, activin regulates SMAD2/3 pathway, and you can see that there's a significant increase of SMAD2/3 in the bone, the phosphorylation of SMAD2/3 in mice from 2 to 12 months of age. So all of this was very interesting. And final piece of data was that the ARC4 knockout mice, so when we remove ARC4 in mesenchymal progenitor using PRX3, those mice had increased bone for the females, but they were losing bone at the same rate. But interesting, in males, they were seemed to be protected against very early age-related bone loss from 2 to 6 months of age. So all of this together was getting a very interesting, attractive candidate for us to study sort of bone homeostasis and maintenance in the aging skeleton, right? So we got very interested into activin signaling. So we wrote a grant, basically, looking at activin ARC4 binding ligands, so activins and others, basically signaling through ARC4 to activate that SMAD2/3 pathway. And I've summarized this here in this little sketch here. So you have ARC4 binding ligands that, as I said, can bind the receptor, the type 1 with ARC4. And that ARC4 type 1 receptor phosphorylate the SMAD2/3 proteins, right? And so we had the model where we deleted, like I said, ARC4 in the PRX positive cells. So bones no longer have ARC4 basically, and we remove SMAD2/3. And remember, I told you those mice were protected against age-related bone loss for the males, and they had a huge increase in bone mass for the females. So in our grants, we use this data and we say, we want to have a systemic approach because that's what people use in clinics, right? We cannot go in humans and delete a receptor in their bones, so we cannot do this. So then we have to treat them with ARC4FC, which basically capture all of the ligands that would bind to that receptor. So we wrote a grant saying we want to use ARC4FC in mice and look at the effect on bone because we think we're going to be able to increase bone mass and possibly even prevent age-related bone loss. So that was going to be a nice systemic approach, but you know, as you know, funding is difficult to get, and we needed to generate a lot of pilot data and really fast in order to meet the grant deadline, right? So, so that's when I thought I went to Vicky, my postdoctoral mentor, and I was saying, well, we don't have DEXA, so that's a little difficult to get data really fast. I would have to do micro CT, and I would have to sack mice at every time point in order to look at it. The mice that I treat with ARC4FC. So luckily, we ended up getting the DEXA from Cintiqa. So we got the iNSiGHT instrument, and I was able to get this done really fast. And I'm going to go into a little bit of the data here. So what I did is I ordered some C57 black 6 mice from Jax, and at 12 weeks of age, and I started to inject them with ARC4FC for 2 weeks. So I was able to follow them with DEXA. I know it was a very short experiment and sort of suboptimal, but it was a pilot just so that we had preliminary data for our grant. And so like I said, we had the iNSiGHT instrument, and I just wanted to put some of the measurements here and, notify that instrument is 570 pounds. So that's definitely not a portable unit. It is on wheels, so it can move, but it's not as portable, obviously, as the PIXImus. But anyway, it allowed us to get multiple amount of data. So we had a significant weight gain observed with ARC4FC treatment, and that seemed to be coming from lean mass. We saw that by DEXA, where we weren't really affecting fat mass. And then there seemed to be no change in BMD and BMC at the time, a little bit more maybe bone mineral density, and a trend upwards, but the P was exactly 0.05 for the BVTV. And remember, this is a suboptimal dose, only for 2 weeks in like a few mice. So that's definitely not the right experiment to confirm our hypothesis, but this was nice trends that was supporting our proposals. So so that was great, and we ended up securing the funding because the reviewers really thought that those preliminary results were promising. So we currently have this project ongoing right now, and I'm very excited to do this. And again, I just wanted to show this because we've had DEXA and we've had the speed at which I can acquire data. I don't think I would have be able to produce all of those parameters fast enough in order to get it. So that was a huge turn in the lab, and obviously we just got the grant earlier this year, so we're very excited about this. And oh, sorry, just want to say in this project, we're like I said, we're going to treat mice with ARC4FC in different types of mice models. So in different metabolic state and at different age and in aging, obviously, which is considered a chronic disease. And so for that, I have to really look at longitudinal measurements. So DEXA is going to be a key instrument for us for this entire project. So I'm very excited for this. And so in the final part of my talk, I want to just say that we compared a little bit of the different systems. So I've talked to you guys about the PIXImus, and now I've talked quickly about the iNSiGHT because we just got it at HSDM. And there's another machine that I want to talk a little bit about, the analyzer. We do not have it at HSDM, but we have collaborators that have it in the Boston area. So that's why I wanted to have it here. And as you can see, appreciate here, the reason I put those pictures is, as you can see, the two newer models are much bigger than the other. So that would, you know, make it a lot less logistically movable into a facility. However, as you can see, they come fully shielded. So that really reduces, well, that prevents all radiation exposure. So that's why I was able to get the DEXA set up in the mouse room so easily and with very little sort of paperwork to do, and do acquire my data really fast, because, you know, we don't have to have a secure location. We've only me in the room. As the PIXImus is open, it is a low dose of radiation, but you do need to be a certain number of feet away from the machine when it's running, because it is not fully shielded. So although those machines, the new one, the iNSiGHT analyzer, are a little bulkier and much heavier than the PIXImus, they offer the fully shielded cabinet, which improves on that aspect. And so I had a collaborator, Jen Coulomb, who came to me. She's from Mary Boxan's group at MGH and Beth Israel Deaconess Medical Center. And so she learned that we obtained the iNSiGHT. And again, like I told you, these guys have the analyzer. So she had this great idea of comparing all of the system to, she's an engineer, so she really has a mind for this. So so she compared all of the system in terms of their variability and things like this. So what she did is she raised a cohort of mice and she sacrificed them, and she contacted me so that we could perform iNSiGHT measurement really fast after death, so without fixing any tissue. And so she came in the morning, I will measure for the iNSiGHT, and then within a few hours, she will measure on the analyzer, the PIXImus, and the MRI. And then she was, you know, cross-referencing all of those data that she was acquiring. And here, I just put that data up in this table, and I just want to show you that if you look, most data points are very similar across machines, at the exception of the lean mass, which tends to be sort of, if you compare it to the MRI, it's slightly sort of under valued for the analyzer, while the iNSiGHT is valued at very similar with data point than the PIXImus and the MRI. But the percent fat tends to be undervalued for the iNSiGHT, although it's somewhat very close and it's overlapping with the MRI results, right? But it is under the PIXImus one. So overall, I would say that in terms of the body composition measurements, those machines are very similar across the parameters. And for the bone parameters, the BMC and the bone mineral density, so the BMD was extremely similar across all of the PIXImus analyzer and iNSiGHT, but we've realized that for bone mineral content, there seemed to be an undervalued value for the iNSiGHT that was significantly different from both the PIXImus and the analyzer. Something that I just want to bring to mind is that the scanning time, we realized, was very different across the machines too. So the shortest one was the iNSiGHT. I could capture and get scans done within seconds. The analyzer, I haven't used it, but Jen told me that it's in terms of it's more on the order of minutes. The PIXImus, I know that it takes a few, like 1 or 2 minutes in order to get the data out. And the MRI, I have no idea, but I know that it's much longer than that. So the iNSiGHT was the fastest equipment to get these results. So knowing this, I just want to say that if you have very large cohorts of mice and you're gonna, you know, it's gonna be very difficult for you to use even something that lasts 2 minutes. It's gonna be a significant use of your time if you're trying to get all that done in a mouse facility. And if you know, like us at Harvard Medical School, you have a very small mass facility with shared room. It becomes extremely difficult because you have to reserve the room, especially for something like the PIXImus, because radiation, there might be radiation exposure, no one else can be there. So all of this taken together, I think is really, I hope is really helping people to pick the right systems for them. And so, yeah, I hope that does this. And Jen is going to publish our paper, I think, really soon. So I hope that's going to be very interesting to you guys. And with that, I just want to acknowledge my current lab. So the Rosen lab, I said, with Vicky Rosen, and the Boxen lab, which was collaborating with us for this comparison study, basically. And with that, I just want to thank you and ask if you have any questions.
Thank you, David, for the presentation. As David mentioned, we will now move on to the Q&A section where Eamon McNeil, product manager at Syntica, will be assisting us. We have a couple questions, and the first one is for you, David.
Mhm.
Are you confident with the lean and fat mass data since that closely matches with the other gold standard of echo MRI?
So I assume that's about the iNSiGHT, right?
Yeah,
Or about the PIXImus data
more
that I got?
more of the data that you acquired.
Yes,
Um,
I would say yes. I am confident with the lean mass and the fat mass data because so far in most of the models I've studied, I obviously don't just rely on that data to make a point. I just go and isolate tissue afterwards, right? So when I finally can sacrifice a mouse. And every single time it was matching what I was seeing in tissue. So I think I gave a very short example here with the fat pads, right? You could appreciate that there was a decrease in fat proportion, and indeed the fat pads were decreased. I've done multiple calorie restriction studies and also high fat diet studies that was affecting the lean mass and the fat mass a lot. And every single time what I was seeing in tissue was reflected by what I saw in DEXA. So I would say that DEXA is actually very powerful to look at lean mass and fat mass. And if anything, I would say it might actually be more powerful than for looking at bone, because unfortunately, DEXA doesn't really appreciate the macroarchitecture of the trabecular cavity, right? The trabecular bone. I showed you guys that here. And so that becomes a little difficult because you could have bone mineral density that is totally similar across your mice, but the mice may have a huge loss in trabecular bone, affecting their bone strength. So I think that's a little, so I would say when you get results about bone mineral density, I wouldn't go and run with it. I would always tend to want to confirm it with microcity afterwards. I hope that's a vague answer to a really good question, but yeah.
I know, really great answer.
Yeah.
The second question is, IGFBP4 causes a reduction in fat mass. What about the effects on lean mass?
Yes, I'm so glad you said that because we did see a little bit of an effect on lean mass, and I didn't go into this because just for the sake of the time of the presentation, but they do have a little bit of a decrease in lean mass as well. Definitely not as strong as the fat mass, and so you can see muscle weight was slightly decreased too. So yeah, there was a little bit of an effect there, but I focused on the fat mass because that's where we had the most, obviously, the strongest effect. And because we're really interested into, like I said, those mesenchymal progenitors, they turn into fat and they turn into bone. So we were not necessarily sort of muscle expert at the time, but we did report those parameters in our papers.
Great, thank you for the answer. The next question is, what are the regional effects on BMC and BMD investigated within the IGFBP4 knockout?
Yeah, so I didn't show the data. I think I just mentioned it really fast. So I showed the aerial BMD and BMC, so the total body results. Like I said, I also gate and draw rectangles on the femoral side specifically. And I didn't look at the vertebra, I just looked at the femoral side, and it was following all the trends of the whole body. So there was decreased bone mineral density, decreased bone mineral content in females, but not in male mice.
Okay, great. Thank you for the answer. The next question is, ALK4 with the FC injection identifies a trend upward in BMD, however, not showing significance. Does that have to do with the dosage or increased treatment regimen longer?
Absolutely. So I think, like I said, this was a very short pilot study to show that we could even just do it without killing the mice, honestly. And so the dose that was reported in the literature was 10 milligram per kilogram of body weight. Here, for the pilot, I had to use 3 because this compound literally cost thousands of dollars. So because we didn't have the funding at the time, I could only do the pilot with this. In order to do it on at least 3 mice, I had to do a suboptimal dose. So that's why I think all of those are trends and they're exciting trends, but they're not, they didn't reach significance. Also, we didn't have a very low N for our mice. So it really is pilot data at this point. I would not necessarily think that this is like, results that you can hang your hat on.
Okay, thank you for responding to that question.
Mhm.
So we have reached the end of our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we'll be sure to answer any questions in the written transcript, and we'll work to get this out over the next few days. I would like to thank David for the great presentation today, and I trust that we have been able to provide you with a better understanding of the latest dual X-ray absorptiometry advances. If in the coming days and weeks you have more questions about the modalities discussed today, I encourage you to contact us here at Syntica, and we'll be more than happy to discuss them further. Thanks again to all of you for taking the time out of your day to attend our session, and we look forward to seeing you at a future Syntica event. Have a great day.
Thank you.
Thank you, David.